Hola, ¿cómo están? Bienvenidos y bienvenidas a una nueva actividad del Festival de Ciencia Puerto de Ideas Antofagasta 2021, presentado con Escondida BHP junto a la Fundación Puerto de Ideas. Mi nombre es Francisco Aravena, yo soy periodista y a continuación vamos a tener una conversación con el físico alemán Erwin Neher, es Premio Nobel de Medicina en 1991 y vamos a hablar sobre la ciencia básica y el rol que cumple la curiosidad como motor de las investigaciones científicas. Y cómo también, gracias a esta curiosidad, él ha desarrollado obviamente su exitosísima carrera, llegando al descubrimiento de la función de los canales iónicos individuales de la célula y el desarrollo de una técnica para medir esa actividad, lo que ha servido de base, entre otras cosas, para el desarrollo de una serie y muchísimos medicamentos que hoy consumimos todos o la gran mayoría cotidianamente. Así que es un trabajo tremendamente trascendente para todos nosotros. Esperamos que disfruten esta actividad. Professor Neher, how are you? Thank you very much for being here.
Fine, fine.
Virtual,
Yeah.
but this is the way we are together now, right?
Yes, I was hoping to come to Antofagasta in person, and to see all these specialties of your area, but unfortunately, that didn't materialize. Yeah. Mhm.
But you've been in Chile a
I've
couple
been
of
in
times
Chile
before.
several times.
Yes,
Yes, yeah.
yes.
Mhm.
How has your COVID life been?
Well, in fact, I got my first shot yesterday.
Wow.
So Germany is a little bit behind in that, I understand that in Chile you have already vaccinated about half the population. In Germany, it's more like 10 to 15%. But I was lucky to get a shot yesterday and expecting to have another one in 3 months, But otherwise, my life was pretty normal, except that no traveling, I mean, before COVID, I used to travel quite a
Yeah.
bit, maybe 4 or 5 times long distance travels in a year. But actually, I haven't been out of my hometown except for once since mid-January last year.
Mm-hmm, mm-hmm. And are you surprised by the speed of developing of the vaccines for COVID?
Yes, definitely. I mean, it's a completely new approach, these RNA vaccines, and there was no guarantee that it would work. Of course, some companies also went on the traditional route, and one could expect that something will happen within some time, but that these RNA-based vaccines are going on the market so rapidly, this was definitely not expected.
Mm-hmm. And do you think there's something to learn about, for instance, the way that the pharmaceutical industry works and the state-sponsored science worked in this case? Should we get lessons in that regard out of this whole thing?
Well, I think that the scientific community proved to be surprisingly flexible in redirecting efforts towards this immediately arising problem, And I was worried that people who are obliged to fulfill the promises, the milestones of their grants would be hindered by that, but I haven't heard about problems. And so it seems that not only scientists, but also granting agencies are quite flexible, or proved to be quite flexible in this response. And I mean, the outcome was indeed very good, fantastic, in the sense that within a short time, there were vaccines available. This on the background that for many other diseases, like HIV or the other SARS viruses, we don't have a vaccine yet, although there have been years passing since these viruses first appeared.
And how do you think we should transform this this whole thing of COVID and vaccine development, science and and science and public policy? How can we make this a teaching opportunity for kids regarding science?
Yeah, well, I think one can make a good story, out of the virus suddenly arising, how it spread, how there was a challenge coming up, and how then rapid vaccine development together with testing, in the end defeated the virus. We hope,
Yeah, we hope.
Yeah. And of course, the story will be the better, the more successful this campaign is. But regarding other aspects, I think an important lesson is that we need to think deeply about the long delays involved in regulatory processes, clinical tests, which of course have to be done, but which take a lot of time, and particularly with the new RNA-based vaccines, I think from the scientific point, it is easy to match mutations of the virus, which is a big problem, that the virus under the influence of existing vaccines mutates and escapes, so-called immune escape, multiplies, and then the science and the governments must be able to respond fast to bring new adapted vaccines on the market. And the science of just changing the virus, the vaccine to cope with mutations of the virus is very easy, because you know what the mutations are, which give an advantage to the virus, and you can just program this into the RNA, which then is the active ingredient in the vaccine. But what's really a bottleneck, or what will turn out to be a bottleneck, is then the regulatory procedure until such a new vaccine comes on the market. I mean, this is what I hear from colleagues. I'm not an expert in vaccination, but of course I have a kind of general overview of biological sciences.
Sure. I wanted to take you back to your beginnings. This talk is purported to be around a curiosity and the importance of that basic element or basic sense in a way to develop science and scientific careers. Do you have a recollection, a personal recollection of how did your curiosity, got awakened, got fostered in the beginnings?
Well, even as a high school kid, I was very interested in both biological phenomena, and also in technical things. And as a young kid, I was taking apart clocks and radios and so on, and was very interested in all these electrical devices, particular. And one thing which then fascinated me was to learn that actually there is electricity in our body, the nerve impulses, which are the major ways of communication within the nervous system. And so at one point, I decided that I want to become a biophysicist, a scientist who studies this electrical phenomena in the body in order to learn more about it.
It's interesting that you choose your what would be the focus of your research, of your scientific career so early on?
Well, it just happened that I got hooked up on this problem, you know, and of course, when then switching to university, I had to decide what I should study. I mean, I wanted to become knowledgeable in the combination of physics and biology, which typically is called biophysics, you know, but at that time, there was no interdisciplinary research education program at universities. You had to study one of the classical disciplines, either physics or biology. And I had to make the decision and decided to first study physics as a basis, but then make an effort to switch into biophysics, biological research as soon as possible. And so in a way, this was a very clear-cut path which I was trying to go, and it turned out fine.
And what in particular about electricity and electricity in human beings fascinated so much?
Well, just the fact that, I mean, as a say when you deal with electrical equipment, you are used to instruments where there are wires, where there are resistors, where there are transistors, you know, and that something like this can happen in the body where there is lots of liquid and well, no metal at all, was a fascinating thing, you know. Of course, this was a problem which had captured the fantasy of scientists since the time of Galvani and Volta in Italy, you know, and even before researchers in the Netherlands and other places had developed ideas about this phenomenon of bioelectricity.
But when you started communicating this to your teachers, your masters, what kind of reaction did you get in terms of getting into this field that, correct me if I'm wrong, but there had been a very, you know, a long history of discoveries, but it wasn't a fully developed field, right?
Oh, I think, I mean, what's called electrophysiology, you know, had a good development in the 1950s, 60s, 70s. The basic understanding of the nerve action potential came with the work in 1952 of British physiologists Hodgkin and Huxley, who were able to show by experiments on the squid giant axon. I mean, the squid has a very large, very thick nerve fiber which runs over much of the body. And you can cut this out and can put even wires longitudinally into this tube-like structure, then apply voltages, measure currents, and so on. And this way they found out that actually the nerve action potential is due to a sequence of permeability changes, that you have this cylinder surrounded by a membrane, and they found out that this membrane becomes permeable to certain ions. First, the sodium ions, which rush in, make the internal positive, and then to potassium ions, which rush out to reverse the thing. And this was a macroscopic understanding of how the nerve action potential comes about. The big problem when I entered the field was, what is the molecular basis? Why does the membrane become permeable? Why does it suddenly let ions pass, you know? And there was this idea already put forward by Hodgkin and Huxley that there might be pores or gates in the membrane which open and close in response to voltage changes. And during my thesis work, this question was very much brought up and put forward. And we then decided, after finishing PhD work, to prove this concept of pores opening and closing, gates opening and closing, by trying to refine the current measurement so that we should be able to see step-like changes in current when these pores open and close. And this was an idea which again fascinated me, together with my friend Bert Sakmann, who at this time was also doing in parallel with my own thesis work, his thesis work at the same institute. And when we later met again here in Göttingen, we decided to tackle this, and in the end were successful.
Well, successful is a way to say it. Let's say that you were awarded the Nobel Prize for that.
Yeah. But it was a lot of luck,
Yeah.
Of course, we had an idea what might work, but it was by far not certain that it would work.
And do you remember a specific eureka moment in that development?
Yes. There were maybe 2 or 3 of such moments, The first, when we tried to record these discrete changes, with a lot of success, a lot of problems. But when suddenly indications of such step-like changes appeared in the noise of the recording, and this was a period when we just gradually improved the conditions so that these signals became clearer and clearer. But they were by far perfect, because in the beginning the measuring conditions were bad. But then there was one eureka moment when suddenly, by chance, the recording conditions became better by a factor of 100 or so that the noise was reduced by order of magnitude. And we saw this very clear up and down in the current.
And when you are moving forward in this investigation and in this project, I'm interested in how would you describe your interaction with the non-scientific world when you are so involved in scientific research, and people from the outside ask you, what do you do, or what are you working on? Did you have like a shorthand for that kind of answers, or did you try to enlighten them because I'm asking because it's something that you need to understand a basic couple of things before appreciating the depth of it.
Yeah. Well, I think, we are in a good position because this concept of these gates opening and closing is a very simple one. You can tell it in a way that people think, why did they get the Nobel Prize for that? It's clear, you just have to improve your recording a little bit and put a pipette onto a cell, and that's it. But of course, there are problems, technical problems. There are many things we tried out and didn't work, and as I said before, this event which actually revealed the signals in all their beauty, to say, was a chance observation that just a combination of different conditions during the measurement make the membrane, which have these gates, to merge with the recording pipette, so that there is a tight kind of seal from which, or a patch from which one can measure them in high resolution. It was a chance observation. And in other cases also we had chance observations which we didn't understand right away, you know, but which shows that as a scientist, you not only should look for the result you expected, but you also should have the ability to recognize that there is something special, which is not an artifact, but which may tell you something interesting. You know?
Could you tell us the story of your partnership with Bert Sakmann? You named him a couple of minutes ago, but he's of course your research partner who shared the Nobel Prize with you. But I wanted to ask you how important was to develop a partnership, and how important was to meet other researchers in other parts of the world who were working in the same field for your own work and research.
Well, I mentioned already that Bert and myself we did our PhD work in the same institute in parallel, and Bert is a medical doctor and studied medicine, and was actually interested from the beginning in the more higher functions, you know, of the brain, particularly details of the visual perception and so on. But he often came up to my lab, you know, because he was very interested in these basic things also, and looked at my recordings. I was, in my own thesis, I was working on snail neurons from the garden snail, you know. And we discussed things and already brought up this problem: what is the basis of the probability changes? Can one prove that there are these gates? And we then went different paths. Bert went to London to join the laboratory of Sir Bernard Katz. You know, I went from Munich to Göttingen to learn about high resolution current recording, because there were labs here at this institute which used novel techniques of high resolution recording. And I worked for some time on so-called artificial membranes, lipid membranes, which simulate what's the membrane of cells, you know. And then it happened that Bert came back from London and joined our institute in Göttingen also, you know. And when he came, we started discussing, and it was soon quite clear that Bert had learned in London to record in great detail from muscle, from the neuromuscular junction, you know, where the transmitter acetylcholine opens ion channels in the muscle membrane. And it became clear that this was the preparation, this was the kind of system on which we might be able to solve our problem. And so we decided to collaborate. He brought in the expertise from biology, the preparation. I brought in my expertise on electrical engineering, so to speak, and high resolution recording, and the combination of these two things was actually one of the keys to success, I would think. You know, I mean, I'm not sure what would have happened if I had been alone. I would never have used the neuromuscular junction. I mean, later it turned out that neuromuscular junction actually was a very difficult preparation, that it's much easier to just take a cultured cell, put a pipette onto it, and record ion channels. But of course, this we didn't know. But you asked more generally also about the connection to scientific community. And I must say, I was very lucky in the sense that my PhD supervisor, Hans-Dieter Lux, was very well connected, both in the German physiological scene and also with colleagues in the US. Before I joined his lab, he had spent 1 year at the NIH. And so he had many friends. And so I was very early then introduced to colleagues like Eric Kandel, who also won a Nobel Prize a few years ago, and other important labs in the US.
Mm-hmm. I wanted to ask you more generally about did you ever have to defend in some way your work in basic science against the pressures of a system that seems to reward more quote unquote productive development, more applied science? Did you ever see yourself in that position?
Oh yes. I mean, it's a constant struggle. I mean, what we do is basic science. Try to find out basic concepts, basic mechanisms of biology. And my work throughout my career has been at the Max Planck Institute, which Max Planck Institutes are dedicated to basic research. And we constantly have to defend our plan, our work towards requests from politics and society to do something which is good for people, And we always have to point out that all the big all the important innovations, applications, breakthrough innovations are all subsequent to new knowledge created by basic science. I mean, no matter what you take, there always has been some basic science preparing the path, so to speak. I mean, if you think of a physical example from physics, quantum physics, which is the basis of all optical devices that we have now, all the computer technology, everything depends on understanding of the quantum nature of light, and the basis of that was laid by Max Planck, who studied the Strahlung, what do you call that in English? The radiation of the black body, which at this time was a very esoteric thing. Why would it be interesting to study what comes out in light or infrared light on a black body, But doing this, he found that he can explain his results only if he assumes that what comes out in energy is in forms of pieces of quanta, of discrete units. So, examples of such things are numerous.
Yeah, sure.
And it's really a constant struggle, But I think, at least in Germany, we are pretty well off because there is a certain understanding of politicians in this issue. Other countries force much more this what's called translation of basic knowledge into medicines and cures and so on. But yeah.
No, please.
Again, I mean, for us, it's relatively easy to argue that the basic things that we did in the 70s and 80s turned out to be an important element for pharmaceutical research today, for developing medicines, for studying the side effects of medicines, for understanding hereditary diseases, the cause of which are mutations in channels, you know, and the importance that this work has obtained in all kinds of biological sciences.
And are you surprised by the applications that your work has had?
Yes, definitely, you know, because when we started, we had no idea of the multiplicity of ion channels and their function, you know. In the 1970s or late 60s, people thought that there were certain classes of so-called electrically excitable cells, like nerves, muscle, and maybe some glands, you know, but there was no idea that channels would be also in other tissue. Today, we know, I mean, through the fact that with our technique, a much wider variety of cell types could be studied and much more possibility there were. We know that basically all cells in our body have a variety of ion channels which regulate many, many things, you see. And so this was a surprise. We didn't expect. We thought, well, we find out about why membrane becomes permeable and understand the electrical signal, but didn't think further, you know, what this might mean. And only with the gradually developing knowledge that there are myriads of different channels and that they're involved in all kinds of normal and pathological function, did this importance of the findings become apparent.
And I wanted to ask you about you are very as a renowned scientist that you are and a Nobel laureate, you're very sought after for giving advice to young researchers, young scientists. What is your main message you always give to them in that regard?
Yeah, the main message is preserve or cultivate your curiosity, you know. Try to find out for yourself what is really the problem which can captivate your mind, you know, which you are able to keep thinking about and looking at this problem from many sides, you know. Once you have found what this problem is, try to get into the best lab worldwide where this problem is being worked on, you know. And of course, it's not easy to get in those labs, you know, but say once you have finished your PhD and choose a postdoc position, if you have done good work in the PhD, it may not be so difficult to get into these labs, but you have to be flexible. You shouldn't look for the best lab in your immediate vicinity, you know. You should really have a international view.
I wanted to ask you about your impressions of science in Chile, given that you have been here a couple of times, and as I understand, you have a working relationship with Ramón Latorre, who works in the same field and is a very important figure in that field also. Do you have elements enough, of course, to form your opinion about the way Chilean science is
Yeah.
developing or not?
Well, I mean, Chilean science has a quite rich tradition, you know. First of all, there is this a marine laboratory north of Valparaiso, you know, which were quite a number of important contributions to the understanding of exactly this squid ion electricity, squid action electricity was done. Are a few famous figures, you know, I mean, Ramon Latorre is one among them. And then I was involved a pretty long time ago in a jury selecting candidates for the presidential awards. I'm not sure whether these still exist, but there was a time when there were these presidential awards. And what became obvious to me is, I mean, at one time I got a box this big, full with application material, you know, out of all fields of science. And what became apparent to me at that time was that Chile is very rich in the sense of quite a number of excellent researchers which were in exile during the time of the dictatorship in the States, you know, and then came back with all their knowledge and expertise and brought Chilean science up, you know. So these two effects, I mean, on one hand, this early interest by researchers in electrical excitability and this inflow of talent, I think gave Chile a good position. Mm-hmm. And I mean, I haven't been now in Chile for number 5 or 6 years, you know, but I think that this is going on in the new institute, which was created in Valparaiso. Mm. I also have been in Valdivia, very interesting place.
Yeah, sure.
Yeah. So, from my point of view, Chilean science is in a good path. You know.
Speaking about curiosity, do you still have burning questions inside of you? Questions that you know,
yeah.
dream about? I don't know, solving yourself, or being a witness to the solving of those questions in your lifetime.
Yeah. Well, I mean, I'm now 77 years old, and I must admit that this kind of captivation, which I was talking about, you know, which happened earlier, every 1 or 2 years, some new problem came up, which really made me curious, you know. That doesn't happen so much anymore. You know? It's more that I have the impression that I have now worked on a certain problem over the last 10, 15 years with quite traditional means, you know, and I just have to finish up that, you know, to focus on that, to get it out in a way that maybe changes the field a little bit, you know, but the kind of drive is reducing at my age, which, well,
and of course there's the age thing, but there's also the Nobel Prize thing, right? I imagine the Nobel Prize made you travel a lot more, giving more conferences, and did that get you away from your lab?
Yeah, well, I mean, this is something I've been asked many times, how did Nobel Prize change your life? And my answer has always been, of course, with the Nobel Prize, you get much more attention, you get much more distraction, because some people think that a Nobel laureate knows everything, is good for everything, is a good politician to talk to politicians, is a good person to fight for animal experimentation, and so on. So a number of things come up and take away some of your time. But you have to learn and adjust to this, and concentrate on a few things that still allow you to have your research going on. But I must say that particularly, say around 2000, I was engaged a lot in Brussels in European research policy, which did take some of the power of my research, But more recently, I realized that actually the time when the Nobel Prize really changes your life is when you get older, because I'm still here sitting in my office doing my research, and this wouldn't be the case if I hadn't gotten the Nobel Prize. More or less, the Nobel Prize protects you, that nobody dares to throw you out, to send you into retirement,
Sure. Mm-hmm. Well, Professor Erwin Neher, thank you so much for this conversation and for this virtual attendance to Puerto de Ideas. And we hope to have you here in the old school way whenever it's possible.
I definitely hope that this will happen, and not too far future.
Yeah, let's hope so.
Thank you. I think it was a very pleasant conversation.
Same for me. Thank you.
Bye-bye.
Y a ustedes les queremos dar las gracias por habernos acompañado en esta actividad, y los dejamos invitados a que no se pierdan ninguna de las actividades que Puerto de Ideas Antofagasta nos trae este fin de semana. Que les vaya muy bien.